All right, welcome everyone, and I think we're good to go. The participants continue to climb, but in the spirit of this being an early morning for most in the U.S., good afternoon to those in Europe or Australia that have joined. Today, I would like to introduce our topic for our second webinar. Let me forward my slide here. Radiopharmaceutical clinical trials and what people should know. So joining me today, you can see on screen, and I'll go through a full introduction in just a second, is Kathy Spencer-Pike, Chief Commercial Officer at Nucleus RadioPharma, and Dr. Oliver Sartor, Professor of Oncology and Director of Radiopharmaceutical Clinical Trials at Mayo Clinic, and also a Nucleus RadioPharma Scientific Advisor Board co-chair. Just a couple of ground rules and what will be covered today. So from an overview perspective, you can see the title, but I'll be your host, Chad Crookshanks, Senior Director of Marketing at Nucleus RadioPharma. And KSP and Oliver will actually go through radiopharmaceutical clinical trials, answer questions around what patients may have, types of trials from a trial perspective, enrollment and participation. So a broad ranging overview of radiopharmaceutical clinical trials. Just a couple ground rules to go through as you heard it start. This audio or this webinar is being recorded. It will be available on the Nucleus LinkedIn site and also available, will be sent out through email for all the registrants. And second, we will have a Q&A session at the end. I would ask anyone to have a question or anyone that has a question to use the chat function within Zoom. And as we get to the end, I will open it up and ask the questions to either KSP or Oliver that you may have. With that being said, I'll introduce our two panelists. Oliver Sartor, as I said before, Professor of Oncology, Director of Radiopharmaceutical Clinical Trials at Mayo Clinic. Dr. Sartor has served as PI or co-PI on multiple pivotal phase 3 trials, leading to FDA approvals on several prostate cancer therapies, including samarium-153, radium-223, cabazitaxel, lutetium-177, PSMA-617. Dr. Sartor has broad expertise in prostate cancer research, extending over a 30-year career, and has published over 500 manuscripts in peer-reviewed literature, the vast majority of which focus on clinical and translational studies in prostate cancer. At last count, his manuscripts have been cited over 46,000 times in peer-reviewed literature. In addition, Dr. Sartor continues to co-lead several phase 3 pivotal trials, including the PSMA addition study and also the NCI-NRG cooperative group NRG-009 trial. Furthermore, he is deeply involved in development of novel therapeutics at levels of multiple other phase 1 and 2 clinical trials. And then Kathy Spencer-Pike, Chief Commercial Officer, Nucleus RadioPharma. KSP brings more than 2 decades of experience with highly matrix Fortune 50 environments, as well as fast-paced growth and startup enterprise. Before Nucleus, she was Chief Sales Officer at McKesson, where she spurred significant company growth, built a high-performance commercial team, and merged 3 legacy organizations into a single commercial organization, achieving unprecedented double-digit growth year over year. KSP previously was VP Commercial Leader at Novo Nordisk, successfully leading the planning, development, execution, and market and sales strategies, exceeding revenue goals. In addition to her commercial experience, Ms. Spencer-Pike is dedicated advocate for DEI, leading the largest US-based women's employee resource group at Pfizer and Novo Nordisk, both of which were the largest ERGs within the companies. And KSP, I think I'm ready to hand it over just to make sure you all can see the slides.
Yes, we can. Yes, we can.
All right.
And so thank you, Chad, and good morning and good afternoon, everyone. And Chad, appreciate the nice tee up to Oliver and I. It is fantastic to be here in Oliver's office at the Mayo Clinic up here in Rochester, Minnesota. There is no snow on the ground yet, and happy October, 1st of October. Wow, the year's going fast. So I'm really happy to be here talking about this particular topic. And you know, it's really intended to increase awareness, just what the slide is saying, engagement and participation around clinical trials. But Oliver, before we delve more deeply into the slides and to the topic, can you perhaps level set the term theranostics for our audience?
Yeah, so theranostics is a term that's been kicking around, gosh, for multiple decades now. And it kind of fuses the therapies and diagnostics into a single word. And the kind of cool thing about a lot of the radiopharmaceuticals is that we can see it and we can treat it. And so on the see it side, we actually have a diagnostic component where we can label a molecule with some type of isotope that can give a signal that can be detected in something like a PET scan or a SPECT scan. And then on the therapy side, we're going to be putting on an isotope that might be able to treat with, say, a beta emitter or an alpha emitter. And then the cool thing is that we can run the diagnostic scan and see where the therapy is going to occur. And this concept of kind of combining the imaging, the diagnostic with the therapy is called theranostics. So there you go.
No. Beautiful. And on this next slide, I think Chad has, it shows a really nice illustration, I think, Chad, for this next slide. Exactly. So when I look at this slide, so you were talking about theranostics. So on the left side of this slide, it looks like, to your point, alpha, beta, gamma, and positron. There's a tremendous number of clinical trials across the imaging diagnostic side and the therapy side. Would you say one is outpacing the other? Would you say the imaging is outpacing maybe the therapy side for clinical trials?
Well, when we look at this KSP, a lot of times we need to establish the imaging before we can establish the therapy. And so there's a lot of products that we're kind of understanding target engagement. And oftentimes, rather than start with a therapy, that we start with an image because we want to understand, okay, where does this molecule go? Where does it bind? What kind of tumors does it find? And that's going to be more on the imaging side. So a lot of people kind of lead on the imaging before they get to the therapy. The other thing is therapy trials are expensive.
Mm.
I can inject an isotope, a diagnostic isotope, into a patient, and an hour later, I could potentially be able to get an image.
Mm.
But I cannot inject an isotope into a patient and understand what's going to happen therapeutically an hour later. That may take months or hell, it might even take years.
Mhm.
So, in terms of like speed of execution, in terms of kind of looking at this from a broad perspective, there are a few that will lead with the therapy, but most are going to lead with the diagnostics. Get a quick, rapid readout, establish the distribution of your ligand, understand the distribution of your isotope, and that allows you then to sort of proceed in a well-informed way on your therapeutic pathway. But please remember that the diagnostics can pay dividends even without a therapeutic being attached. I mean, what do you think we do for all this staging? We do breast cancer, prostate cancer. We run a PSMA PET scan for staging of prostate cancer. Well, not all those patients go on to therapy. We just want to know where the cancer is.
Right.
And that diagnostic side is really, really important. And I want to keep that in focus in addition to the theranostic. Sometimes it's just about the diagnostic.
Yeah.
That's important too.
Okay, excellent. Well, and that kind of takes me to the right side of this trial. So when you look at the top cancers by trial, prostate clearly outpaces the rest in the radio pharmacy side. Why is that?
Well, there are a couple of reasons. Number one is we have kind of the rise of the diagnostics. And if we want to look kind of at the two leaders in the field, I'm actually going to cite a relatively rare tumor, the neuroendocrines.
Okay.
And that has a receptor, the SSTR2 receptor, that's somatostatin receptor type 2. And that's a really cool and interesting tumor that benefits from a theranostic approach. It's pretty rare. I mean, it might be 5,000 or so a year. Well, remember for a moment, I'm going to give you some statistics.
Okay.
300,000 men diagnosed with prostate cancer this year in the United States. 300,000. It is a lot of people.
Yeah.
And then with the discovery of PSMA a while back and the development of PSMA imaging, now we have a tool that really works. Well, there's a lot of interest in this tumor because there's so many different ways to do it. It's a big commercial area. And then, of course, in the therapy side, we have the rise of PSMA 617 lutetium, brand name Pluvicto, which is a very impactful new treatment, currently selling over $1 billion a year for our Novartis. And that has really been kind of a jolt forward for the field. Yes, there was the SSTR2 therapy, very important for the neuroendocrines, but it was a tiny little field. And if you're working in a tiny little field, you don't generate the same degree of excitement if you have a big, important neoplasm that affects so many people around the globe. And now you have both a diagnostic and a therapeutic to take advantage of this approach. And it's just like a snowball. It's just boom, it just gathers and gets bigger and bigger. And now people want to do it better and they want to take different variations. And by the way, there might be different dosing and there can be different combinations. There can be different alpha beta isotopes. All of a sudden you just have a just flowering of this exploration. So it's been a remarkably fun field to be a part of. And, I'm a prostate guy for a long time. And to watch this explosion, very cool.
Yeah, well, and as I'm sure everyone listening, those of us in the room, I mean, everyone seems to either know somebody or is somebody who's been impacted by one of those cancers, and to see the innovation happening across the other cancer targets has got to be incredibly exciting as a provider.
Just a little bit of a comment, KSP. We look at prostate at the top, and that's where I emphasize. But look down the list a little bit. Okay, number 3 on the list is this neuroendocrine, this rare tumor. But now we have kind of a proven approach. What we'd love to do, and I think everyone in the field would love to do, is extend beyond the prostate and the neuroendocrine. We do want the breast, we want the lung, we want the colon, we want the pancreas, all these other big tumors to benefit from this approach. That's going to require different targets. It's going to require maybe different isotopes, maybe different combinations. So we've our work cut out for us. We have a big worthy foe in human cancer, and it's not all the same. It's probably 110 different diseases or more. So each one of them is going to take a little bit of an individualized approach.
A little bit of a secondary question to one of the things you said, receptors, and with the PSMA, trying not to get too far over my skis and asking a technical question. Is prostate a bit easier to go after because of the receptors compared to some of the other cancer targets?
I don't know if I would say that. We got a little bit lucky on the PSMA. We have a lot of expression. But actually, I've used the word PSMA like 5 times already. Prostate specific membrane antigen, just so in case you're wondering. So it came across as prostate specific membrane. And that was really good. It actually is not completely specific to the prostate, but few things really are. But it was highly expressed in prostate. It gave an exploitable target. Now, a lot of other tumors have potentially exploitable targets, but then we really have to develop the proper ligands and isotopes to really go after it. And then the other thing, and this is something that sort of befuddles people who are not in the field, you also have to understand the therapeutic landscape. Imagine for a brief moment that you have something like myeloma, where we're currently getting complete remissions with the CAR T cells. But we may not need to target the myeloma where we already have an effective therapy. We have to understand the therapeutic landscape and understand the needs of the patient, and then to be able to bring your novel therapies into those needs in an impactful way. So you have to understand not just the target and the isotopes, you also have to understand the tumor and the therapies available for that tumor in order to provide an advance. And so there's a little bit of complexity beyond just, oh, here's a target, here's an isotope. The therapeutic landscape is very, very important.
So you just triggered a question for me on that side. So as you look at, say, the ADCs, the CAR Ts, radionuclide trials, of that landscape, do you think there are more challenges or opportunities? I'm asking you to look at your little crystal ball, in our space versus others. In other words, do you think providers and patients would be more apt or less apt to come into a clinical trial where there's radioactivity?
Very good question. And by the way, you mentioned 3 approaches, and I'm even going to put in a 4th into that category. So to some extent, if you have a cell surface target, you might think about the radiopharmaceuticals. You might think about the CAR T cells. You might think about bispecific antibodies, which is another sort of
Yeah.
rising star in the therapeutic armamentarium. And you might think about ADCs, which are antibody drug conjugates. So you really have a multiplicity of ways to approach it. Now, everybody probably has their biases. You know, Pfizer just paid what $40
Oh,
billion to
yes.
take over CGEN, and
Yes.
that was an ADC play. And the CAR Ts are rising, but you know, the CAR Ts are really complex. I'm a little less of a fan of the CAR T cells. They've looked good in the hematologic malignancies, not so good in the solid tumors, but you know, maybe that'll change. And then, you know, the bispecifics is also led in the hematologic malignancies, but now we have a little bit of activity in small cell that's getting people excited. And the ADCs have fanned out across the way. But, you know, I actually have a bias toward the radiopharmaceuticals. And some of the biases are just built around the concept that I can take that isotope, I can put it on the tumor, and then I can have a predictable effect. You know, if I'm putting an alpha emitter in the right place, I'm going to kill that thing. And with the ADCs, there's problems with the linkers and the loose linkers, and then the little toxins that go flying around. I'm a fan of the radiopharmaceuticals. To me, if I were a patient, I want to go to where the activity is. And look, if you've got a bad cancer, you probably want to take a risk. You know, risk of the bad cancer, risk of some therapy. Well, somebody's going to have to make a choice. And I just want to go where the action is. I want to go where I think I'm going to get the best response. And
Yes.
it's hard to take, say, you know, absolute radiopharmaceuticals can be
Sure.
better. I'm not going to do that because it's not really factual. But let's just take an inclusive, all of the above need to be in our armamentarium. I happen to like the radiopharmaceuticals, but that's because I believe that they're going to work if I get them on the right target.
Yeah, that's great. Chad, if you'll flip to the next slide, I think the next slide will drive home a little bit of what we're talking to as well. So we're talking about, just to your point, the radiopharmaceutical landscape. And this is the Oppenheimer view and graphic that has become quite popular across the field, which is fantastic. And it really, just at a glance, shows you just the plethora, the abundance of clinical trials and the different phases, and to your point, the number of players that are in this space. So you just mentioned 4 different, if you will, approaches. How in the world are providers who are not in your space, not med onc, not maybe in the nuke med, how are they supposed to stay on top of this type of innovation? Because the last stat I think I saw was less than 5% of patients diagnosed with cancer enroll or are enrolled. So how are those providers supposed to stay on top of this?
You know, I know what I would say,
Okay.
and it's an honest statement.
Okay.
Perpetual confusion.
Well, yes.
Okay, because
Overwhelming.
you know, I mean, I treat one cancer. I treat that's all I treat. And it turns out that even trying to keep up with prostate is like a really tough thing. And you know, I look on the Oppenheimer wheel, which by the way I really like. It probably needs to be updated every hour. But on the other hand, you know, trying to keep up with this landscape in and of itself is really tough. So when you're a provider as a general oncologist and you have the plethora of information just sort of just smashing you every morning when you wake up and check out your internet, you have to learn how to prioritize. And, you know, I think the good way to prioritize if you're a practitioner is the FDA approvals. You know, just keeping up with the FDA approvals. I don't know how many there were last year, what, 23 in cancer? And just keeping up with that is going to be plenty. So I personally love the New England Journal because the really good stuff kind of rises to the top. It's not to say that important stuff cannot be published outside the New England Journal. Of course, you can have important findings outside the New England Journal, but if it's in the New England Journal, it's probably important. So for me, the FDA approvals, New England Journal is kind of, it's the tip of the iceberg, but if you want to skim it, that's what you need to keep up with. If you want to delve into this field and really sort of get your hands around the nitty gritty of the drug development phase 1, 2, 3, and all these companies, you have to specialize. There's just no other way. It's just an overwhelming amount of information.
So follow-up question to that, Oliver. So if a patient is newly diagnosed with prostate cancer or breast cancer, and they're looking at the number of physicians or oncologists, med-oncs in their particular area, should they look for, because this is news to me and this is, I'm learning as I'm talking to you as well, should they be looking for providers that specialize either in ADCs, CAR T, or does it matter?
I think it's not really practical to get
Yeah.
to the super specialized, right from the beginning. The first thing is you need to kind of sort it out. By the way, let me tell you how to be an oncologist. It's pretty
Okay.
easy, actually.
Go for it.
Okay, so first of all, you need to make the diagnosis.
Okay.
Okay,
All right. Number one.
Yeah, number one. If you don't know what you're treating, you're probably not going to be treating it right. And that typically does require a biopsy. There are a couple of types of tumors that we can treat without a biopsy, but we have to get some tissue in order to understand the biology of the tumor. Then we do something called stage the patient. We have to understand the extent of spread. So a tumor in the prostate and a tumor that's in the prostate and the local lymph nodes, or a prostate tumor that is in the prostate, the local lymph nodes, and the bone, are all three different categories and all kind of require a different approach. But first of all, make a diagnosis. Second of all, stage the patient. Then all you have to do is treat them properly. That's it.
That's it.
That's it.
All right.
That's all you have to do.
That and stay at a Holiday Inn.
Yep,
And
there you go.
Okay, so back to the clinical trials. So it appears that most clinical trials are being held at large academic medical centers, flagship centers, cancer centers. How are patients and providers that are more in the regional community hospitals, how are they ever able to take advantage of clinical trials, or can they?
Well, there are a couple ways. You don't actually have to run clinical trials to benefit from them.
Okay.
First of all, the reason they kind of center in the larger centers as a whole is that you're going to be having a expertise and infrastructure that's required for clinical trials that kind of goes beyond what a typical community center might be able to offer. But the reason we do trials is not to benefit people in the centers, it's to benefit people everywhere. And the important trials that we do are going to impact patients, whether or not they be in New York City or North Dakota. So that's the job of the good clinical trial, is eventually to diffuse those results throughout the country to be able to have everyone benefit. I mean, what's the good of having a therapy that people can't benefit from?
Cool, yeah.
So, if you're in a community setting, you can take advantage of the learnings, even though you may not benefit. And second of all, perhaps you have a difficult patient, you have a complex patient, you've done what you can, then at times a referral into a major center can be beneficial for the patient. And that's why we're here, is trying to help the patient.
Excellent. Super. Thank you. All right. If we go back to this slide, can you spend a couple minutes or a minute or two explaining the little graph to the far left, upper left? We see the peptide antibodies, small molecules. It looks, if you look at the little legend and then you come back to the circle, it looks as if a lot of it is the peptides, the antibodies. Would you say there's a particular trend occurring in the clinical trials?
Let me back up and give a little bit of a context.
Please.
I talked about theranostics
Yes.
in the beginning. And I talked about the isotopic globalization,
Yes.
because what we want is we want to get the isotope to the right spot. Well, how do we do that? We attach it onto something that is going to bind to something on the cancer cell or in that microenvironment. And that's something we're going to call a ligand, or, we can just call it the isotope carrier for a second. But then we can break those down into a bunch of categories. So we can have like really small little molecules. We can have naked molecules, like we can inject radioactive iodine and it gets taken up in thyroid, okay, through something called the symporter. So that's the simplest of all, or radium. It's not even attached to anything. It's a naked isotope, or strontium. So some of these isotopes will naturally home to a certain spot. And then we can have some little molecules, and I'll take the PSMA binders. We can use things like PSMA INT, are the one that commercially is under Polarify. Illuccix, which is a PSMA 11. I'm not trying to be on a branded hinge here. I'm trying to make sure I spread the brands around. But these little molecules that kind of carry the isotope and bind to the PSMA. But now, not everything has got little small binding pockets that you can use these small molecules. sometimes we have something on the cell surface, but we can't target it with a small molecule. Maybe we need an antibody. But let's look at the complexity of antibodies for a second. So we can use like whole antibodies, and that's one approach. And then we can start chopping it into pieces like an FAB fragment. It's a piece of an antibody. Or maybe when you use like a camelid or a variable heavy chain, the normal antibodies have two chains, a light and a heavy, but now they're heavy chain antibodies. And then we can give them in various sizes. And oh, by the way, we can make multiplers, and we can make antibodies with 2 heads and 3 heads and 4 heads. And we can create all these variations. The whole idea, well, and I'm looking up here, I'm getting back to your thing. So what we've got are peptides, antibodies, small molecules. We just engineer chemically the best approach to carry the isotope onto the cell target and hopefully deliver it with the least amount of toxicity. Now, we're going to have developments in every single one of these areas. In fact, as we do today, I mean, let me talk about antibodies and isotopes in prostate cancer. Okay, so we have a new little company called Convergent, and they have an antibody to PSMA, and they're going to be carrying actinium. And then we have Telix is working with an antibody, and they're going to be carrying initially the lutetium and later on an actinium.
YMABs.
And we have, well, YMABs is a whole different beast. Okay, that's a whole,
Yes.
let's not even go there, because
right,
it's like, wow, that's like it's a very cool concept, and I love the YMAB people, but it's like they're unique and they're doing their own thing.
Unicorn.
And then we have something called pre-targeting, where we're actually combining the approach. And I'll just mention what pre-targeting is. First of all, there's things that you can make an antibody to. You can't make a small molecule, so initially you tag it with an antibody, and then you send in the bombers to go bind the antibody. I call them bombers because they're the isotopes. They're going to go bomb. And so you pre-target with the antibody, and then you bind the isotope to the antibody that's bound to the target of the cell. That's called pre-targeting. And it's all the above. YMAB's approach is yet even different again with these self-assembling molecules that will be excreted when they don't self-assemble and bind. And when they self-assemble and bind, then they'll stick. It's a very cool approach. But anyway, taken together, I am not enamored with any one particular approach. I kind of like all the above. I must admit, I'm a little perhaps more biased towards some of the smaller than some of the bigger molecules. And because I like the idea that we can bind something quickly and then we can deliver. By the way, the second portion of this we haven't even touched on. It's the different isotopes. And we have really short-acting isotopes. You're not going to talk about astatine-211,
which has a lot of excitement.
Has a lot of excitement. It's a single alpha,
right? But challenging.
7-hour half-life.
Yes.
We had lead-212, which is also exciting with like a 10-hour half-life. Then you have actinium with a 10-day half-life, and then you have lutetium with a 7-day half-life, and a lot of things that kind of come in between. So we can take our small molecule and put on a whole bunch of different isotopes. We can take our big molecules, a whole different bunch. But the key thing is you want to match the isotope with the molecule you're using for targeting. It doesn't make sense to put astatine on an antibody because the antibody is going to circulate around for a while, and the astatine is firing over a 7-hour period with a half-life, and it's going to be gone by the time you do it. So an antibody is going to need a longer isotope, but something really short that binds quickly, you can use a short-acting isotope. So I'm talking a lot, but I'm trying to give a perspective on the interplay between the isotope and the carrier of the isotope, and then all of these different approaches, and then you see the wheel and you realize, oh my gosh, the complexity is just really high.
Right,
but it's good.
It's really good.
It's really good. This is good stuff.
I mean, this is really good stuff.
Yeah.
So I'm going to get to also the topic of supply chain and infrastructure challenges, which we know have plagued radiopharmaceuticals for years. So when you take the outer ring, of course, of this graph is kind of phase 1 or preclinical, but as those, and let's say God willing, all of these different programs actually advance and are successful, and as they advance and get closer to commercialization, that means we need more isotope supply. So
Absolutely.
do you want to, I mean, is there enough today for what we have? And do you believe we're going to have enough in the future?
Isotope supply is a really interesting question. And, first of all, you've got to be able to do the whole CMC package
Yeah.
for an IND. And that means you've got to have isotopic purity, you have to have manufacturing, even just to be able to get into the people. But having an initial supply and having a commercial supply of a product are totally different things.
Sure.
I can make an isotope in adequate quantity to give to one patient,
Right.
or am I going to try to treat 30,000
right.
patients, and that scale is totally different. And remember, you're selling ripe tomatoes when you're selling isotopes because they're decaying.
Yes.
As soon as you make it, it's actually going away.
Yeah.
So you've got to have all the parameters in place, the quality controls in place. I'm going to simply say that isotopic production and drug manufacturing in the radiopharmaceutical space is a really big deal.
Yeah.
And it's an easy place to stumble. And it's really going to take a village to be able to do this. If I'm a little company and I'm designing the perfect isotope along with a perfect ligand to be able to treat the cancer that is really a problem for my mother-in-law, I'm going to have all my thinking all put in place. But then I have to get the components. Where am I going to get the isotope? I don't manufacture isotopes. That falls into another category. That's a whole different area of expertise is isotopic manufacturing. Then you got to make the drug that you're actually going to give. That's a whole different area. That's what you guys do. I mean, you're going to have to master that part, and then you got to deliver it, and then you got to administer it safely in a proper environment. It takes a village. It really does.
Yeah.
It takes a village.
So I hear you saying, yes, we probably still do have some infrastructure challenges,
Oh, yes.
but a lot of great minds are thinking on it.
I think it's a nice way to put about it. In any new field, we are going to have to press it forward quickly and conceptually in order to bring these advances to life. And the infrastructure that is required is just going to have to somehow occur. Well, somehow occurring just doesn't happen when you wake up at night and have a dream. It takes planning, it takes execution, it takes collaboration, it'll take contractual relationships as you build the isotopic needs, the manufacturing needs, the transportation needs, the clinical needs. It all fits together into a package. And like I said, it does take a village. Everybody has to work together in order to make this work.
Yeah, well, I know I'm incredibly excited, of course, working at Nucleus and as a CDMO, and being on the side of helping these clients and these companies bring their therapies to the market is incredibly exciting. All right, I'm going to switch to timelines. So, with my background having been in big pharma, more the huge chronic disease state medications, very familiar with those clinical trials. What's the timing and how does that compare to clinical trials in this space? So for example, if you take those products or those programs that are sitting in a phase one, how long is a product in phase one or phase two to get to potentially commercialization?
Yeah, that's not so easy to answer because there's a lot of variations. And a lot of it is about the acceleration that you can get with the theranostic approach. First of all, patient selection is a really important thing. It's one of the things I really love about the theranostic approach is we can choose our patients wisely. Somebody didn't express the target, we don't necessarily have to treat that patient. With a typical drug, we just kind of give it to everybody and then accelerate. So I think we can do faster. There's also something that's not on here called a phase 0 study. And a phase 0 is really just an imaging study. We kind of understand a little bit about metabolism. There's a special carve out for the phase 0s. And just a single image can have tremendous value in this field because it tells us, oh, the target's there and we reached the target. So the phase 1 is a little bit hampered because remember that's your dose finding study. And we don't always know the right dose to start. So we have to start low in order to be safe. We kind of extrapolate from some of our preclinical studies. And then we have to escalate up into the dosing range. So the phase 1 is really just kind of a dosing study. We're trying to figure it out. Of course, we're looking for activity too. That can take even a couple of years in phase one.
Okay.
Yeah.
Okay.
Now,
Wow.
what we're
Okay.
increasingly trying to do, or I've seen phase ones last for 3, 4, 5 years, just to let you know. You're looking for activity in the phase one. I mean, if you don't have
Okay.
any activity in your phase one, you probably have a problem. Your phase two is going to be this idea that, okay, we kind of got the dose down. Now we want to see what the activity is in patients. And then the phase three is, of course, the comparison
Yeah.
of your novel agent to a standard of care. And of course, that can take years as well, because oftentimes, like our PSMA addition trial, 1,100 patients, and we recruit it really fast. We have to wait for the events to occur. So that's really important. Picking timelines is a little bit hard, but for phase one, often a couple of years
Okay.
is reasonable. For
Okay.
phase two, it may take a year or two, and then for phase three, it may take another 2 to 4 years. So we're looking at probably, if it's fast, it
Yeah.
might be 7 years. That's fast.
Yeah, that's fast.
Yeah,
Okay.
and but it takes a while to get all of these things sorted out. And don't be discouraged by that, because we're actually getting faster and better all the time. I mean, I look back in the past and I just feel like I know so much more
Mm.
today than what I knew in the past. And I can apply that and I can accelerate the possibility for cure. And I'm excited about that. We're getting better, KSP. We really are. We're getting better here.
That's really encouraging to hear. And you'd mentioned kind of the number of patients. Are the number of patients traditionally seen in a radiopharmaceutical trial less than what you would see in other clinical trials for, cardiovascular disease or diabetes?
For cardiovascular and diabetes, the answer is that oncology typically runs
Yeah.
smaller trials because, for the diabetes trials, often you look at, thousands of
Mhm.
patients, cardiovascular trials, tens of thousands of patients. We can typically get our signals in pretty small trials. I actually was, last night, I was designing, I was helping somebody else design a trial, and I was suggesting 12 patients.
Wow. Okay.
And as an initial, because we kind of know that we know of the approximate dose. And I said, look, let's think about this. If you treat 12 patients and you have no responses, do you really want to treat anymore? I don't.
Mm.
All right, if you have one response, is that a signal to go forward? Not in my book.
Mm.
Two, kind of borderline. Three or more, and this is a refractory population. If I have 3 out of 12, I probably want to do some sort of expansion. But rather than say, hey, I'm going to do 60 patients on day one, I'm really taking a baby step and say, okay, I'm going to do 12, and then I'm going to evaluate. If I'm 0 for 12, to hell with it. I got better things to do.
Yeah. Okay. So now that I am a new oncologist, and I'm trying to wrap my brain around that. So 12, if they stuck with 12, whomever you were talking to, if they stuck with 12, is that statistically significant at the end of the day?
No,
No. Okay.
no.
But it's a
No.
starting point.
I love statistics.
Okay.
And when it comes to phase three, they rule the day. But in the earlier phases, you want to go, no go. By the way, the traditional number is 14.
Okay.
Yeah,
Okay.
because if you're running 0 for 14, you can exclude with like a 90% power the probability that the response rate is actually 20% greater. Okay, that's
Okay.
a statistical approach.
Okay.
You can do that. I'm looking for early signals,
Mm.
and I don't necessarily need all the statistics to be able to bolster that. Some of it to me is common sense. I have been on a trial where I treated 10 patients with no responses. And
Mm.
you know what? I didn't want to put the 11th on.
Sure.
Why,
Yeah.
why do I want to pursue
Yeah.
something that I don't think is helping people? Well, it's not statistically significant, Oliver. You need to get to 14 to be able to rule out that 20%. No.
No.
Look, I'm in the business of treating people,
Mhm.
and I don't want to treat somebody with a drug I don't think is going to work.
Mhm.
So I'm a little more clinically oriented, and I can sort of ride the gestalt. Now, we do have to understand the different patient subsets, because if I treat the wrong 10 patients in the beginning, then okay, that's a different scenario. But I'm talking about patients that I've selected for proper treatment. I know that they express the target, I know the dose, and now I'm kind of bringing it together. On an initial phase one, of course, you're going to do it's very exploratory. So I don't know, take what I say with a little bit of grain of salt, but it takes common sense too. It's not all about the statistics.
Yeah. Wow. Well, and you know, they whoever you were talking to, such fantastic insight and expertise from your vantage point to be able to give them some input on how to conduct clinical trials, how to design clinical trials. Because really the design of the clinical trials really determines the future of that drug.
Absolutely.
Right.
Now, this drug already has a future.
Great.
And so not to give away some of the details, but this drug has a future. But what we were doing is to swing it into a new patient population.
Mm,
And the new patient population is very, very defined. Actually, I'll tell you what it is. It's a post-Pluvicto patient.
mm,
So the experiences come around in kind of the Pluvicto naive space. We know there's activity there, but we don't know anything about the post-Pluvicto space. So let's just run a small trial there and find out, hey,
mhm,
can we end up with this novel method that seems to have a good signal
mhm.
in the Pluvicto naive patients? What about the Pluvicto pretreated patients? Well, we don't know. So let's just run a small trial and figure it out.
Mm.
And, again, it doesn't take a huge number of patients to do that. Now, as I was pointing out this morning to the individual, that there is a little bit of complexity to the post-Pluvicto patient because I distinguish between what I'm going to call the refractory patient and the relapse patient.
Okay.
The refractory patient is a guy that might get the Pluvicto and then blow right through it. They have no response. And the refractory patient is going to be a big challenge. The relapse patient is a patient that has responded. Right now, we're restricted to 6 doses, and then they're relapsing afterwards. So they were sensitive, and now they're relapsing. That's almost a little bit of a different patient population. So, we have to be a little bit careful about how we divide, the post-Pluvicto patient, because refractory and relapse are 2 different categories there.
Good distinction and nuanced a bit.
Yeah.
So when you also then look, so if I'm a patient and I'm looking at all of this, or even a provider, and we know that many of these therapies are indicated, well, Pluvicto right now, we're hoping that change. I know Novartis went for pre-chemo indication. Do you see therapies, radiotherapies, becoming the standard of care before chemo, before other types first?
Yeah, I think it's reasonable to be hopeful about that. Chemotherapy is rarely anybody's favorite. And, it's not that chemotherapy is not important. It is important. And we've used chemotherapy as our initial paradigm. But I think we have to open the door for new paradigms as well. One of the things about, and I'll go back to the PSMA lutetium for a second. There've been comparative studies to look at a phase 2 randomized design coming out of Australia with a chemotherapy, cabazitaxel, a good chemotherapy, and the PSMA lutetium. And in this randomized trial setting that was conducted in the post-chemotherapy, the post-taxane space, but that was the second taxane, the patients who had the isotope did not live longer, but they lived better. And so the side effect profile, the quality of life profile was improved. So at times, if we can get to the same place, but with a better quality of life, that's an improvement too. The 2 parameters that I really pay a lot of attention to, KSP, do you live longer or do you live better?
Absolutely.
Ideally, we want both. But living better is also really, really important.
Yeah.
Suffering the side effects from chemotherapy is not that much fun. If you can get away without the chemotherapy side effects and have the same outcome, that's a victory too.
So, living better, quality of life, typically insurance companies don't pay well or want to pay for that. What do you think the landscape of reimbursement looks like?
I think it's going to be okay if we get the FDA approval.
Okay.
And that's going to be really the key. And the FDA is focused on this as well, as
Great.
and the safety too. And I want to mention something about safety that is a bit of an unknown with the isotopes we need to know more about. But, if you're designing a trial and you help somebody live longer, then that's an easy sort of endpoint to understand. The living better part is going to take a little more analysis, and there are standardized methodologies with health-related quality of life, adverse events that need to be looked at. Let me mention one of the bugaboos about potentially bringing the isotopes a little bit earlier. So isotopes can be associated with longer-term deleterious effects, just like chemotherapy.
Okay.
Okay, so imagine for a second that I'm giving a compound
Mhm.
that may actually be causing leukemia down the line,
Mm.
7 and 8 years later. Okay, well, there are chemotherapies that do that. Okay, and they can cause myelodysplasia and other such things. So we have a bit of an unknown
Mhm.
about the isotopes in terms of their long-term follow-up. And I think it's really imperative that as we do these studies, that long-term follow-up be incorporated as a mindset at the very beginning, even though the patients may not be going to be living 3, 4, 5, 6, 7 years, particularly a lot of these advanced patients. We still need to be committed to following these
Yeah.
patients to understand any long-term adverse events that might occur, because that's a question in the field. And the
Mhm.
FDA is going to mandate that, by the way.
Okay.
And
Good.
yeah, I mean,
Good.
like for the radium trials,
Yeah.
there's a registry called the Reassure Registry. We're following thousands of patients that have been treated with radium all the way through as long as possible to look for any deleterious
Mm.
effects, second
Mhm.
malignancies, myelodysplasias, problems that can occur from long-term isotopes. You need to commit to that if you're in
Mhm.
this field, because it's an unknown question, and we really need to take the unknown out of the equation.
Absolutely. Again, it's the trade-offs, right?
Yeah,
Yeah, definitely.
absolutely.
Chad, I see that there may be two questions. I'm not sure if we are unable to see. I thought maybe you could read them out to us, Chad, if there's questions.
Let me come back up here. There we go. So I have three questions.
Excellent.
And one I think we can stay on the Oppenheimer wheel here. Was what was the most advanced clinical trials in the RLT space?
Yeah, the phase threes are the most advanced trials.
Which
Uh,
I would say is the blue. If you
oh,
look at
oh,
yeah.
I'm sorry. I've got to take my glass off and look at it closely. Um, yeah, in the middle of the wheel are the actually what is the middle of the wheel? Oh, those
Yeah,
the,
Pluvicto,
yeah,
Lutathera.
what the middle of the target is approved.
Yes.
And then before you get to approval, you're going to get to the phase threes. And then as you come out, you have the phase twos and the phase ones, and then the preclinical stuff. So, I mean, some of those, like the SPLASH trial, which is PSMA INT PNT 2002. I just presented at ESMO when two weeks ago. We're waiting to hear from the Eclipse PSMA INT from Curium. So that's a phase three we haven't heard from. We have a phase three that'll be analyzed sometime, called the PSMA addition trial. Accrual is completed. This is in the hormone sensitive space. And then it's hard for me to see because it's written in little tiny things. The TLX 591 phase three is now
Yeah.
is now launched. That's a PSMA antibody with lutetium. So that one is now launched. So, if you're trying to keep up with the field, you have to keep track of all these. And the phase threes are well publicized, but then as you work your way out the wheel, the phase ones, understanding where people
Yeah.
are, that becomes a little more of an artistic sort of understanding. And oftentimes it's not even completely clear.
No. Yeah. Hopefully that answers that person's question. If not, hit us back with another question. Chad, any more?
Yeah. Another is the risks associated with radiopharmaceuticals versus other. I think you talked a lot about the, difference with the side effects versus chemotherapy, but the question was, any risks associated with radiopharmaceuticals.
Sure.
Yeah, and I made an allusion to that with the necessity for long-term follow-up. We are giving something that can damage the DNA of normal cells. That's called a mutation-making machine. And mutations can cause secondary cancers and other deleterious effects. And some of these are quite frankly unknown. Now, if the patient's going to live for 6 months, it's probably not much of a question. If the patient's going to be living for 6 years, it is a question. So, understanding the long-term potential deleterious effects of radiation is an important part of the equation in this field. It may be a little less important, say, with hormonal therapies and the like. So, yes, there are dangers that occur, but it's all about the risk benefit ratio. And, just imagine for a second that if I have an incurable cancer, my life expectancy is 1 year, and I get treated with one of the isotopes, and now I live 6 years, and then I have a deleterious effect 6 years later. I mean, I hate to say it, that's still a victory because I managed to live 6 years, whereas otherwise I would have died. So, but all of these have to be taken into account. In the end, it's risk benefit. In the end, it's about informed decision making. And in the end, it's also about understanding to the best of our abilities the data regarding the risk and benefits and gathering the data over time that's necessary to understand that calculation.
And maybe Oliver, you can speak really briefly to, I think it's helpful for maybe to the person who asked that question and maybe others is you hear like chemotherapy is a systemic therapy, but there's the word precision medicine, precision therapy used a lot with RLT. Can you speak a little bit briefly to what's the difference?
Yeah, so precision medicine in general is about targeting a specific group of hopefully susceptible individuals to that particular therapy or susceptible cancers to that therapy. It's not something we use to treat everybody, but we can find the right patients. Let's go with the PARP inhibitors as a good example. So, the PARP inhibitors are an interesting way of kind of exploiting DNA damage repair. People who have a defective DNA damage repair pathway, say a BRCA2 mutation, are particularly susceptible to the PARP inhibitor. And the normal individual is sort of like kind of plus minus, but the individual with an underlying BRCA2 mutation is uniquely susceptible to PARP inhibitors for those particular tumors. So that is an example of precision medicine. Here, it's another type of precision medicine. We're looking at the PSMA expression on the cell surface. I can do an image, a PET scan, and I can say, you got it, you don't. Now, I'm not going to treat the patient that doesn't have the target. If you express a lot of the target, I think it can be very good. So I can precisely use my therapy in that patient with a lot of PSMA PET expression. And that's precision medicine to me. I'm precisely picking the patient that I think is most likely to benefit.
Excellent. Thank you for that. All right, Chad, I think there was one other question. Yes.
Yeah, there's one more, and it's in regards to your comment on the 5% patients enrolling in clinical trials. So, Dr. Sartor, do you see a difference in patients' excitement to enter a targeted trial? And I mean, just you were talking about it there with PARP inhibitors. Are patients more excited to enter a targeted trial versus a chemotherapy or non-targeted traditional trial?
I think so. Because we're a little bit selective in who we treat. When I go through the discussions with the patient, and particularly if I'm at the phase 2, phase 3 level, I'll say, look, I'm going to try to understand if you express this particular target. Now, the bad news is that if you don't, this trial is not for you. But if you do, then this trial could be for you. And the idea that somebody is going to undertake and spend some time thinking about the individual cancer and the expression on the surface of those cancer cells is enlightening to patients. I say, look, you express a lot of PSMA, I can target you. Or unfortunately, I could do a scan and say, I don't see the target. I'm not going to be able to treat you on this trial because I don't think you're going to respond. So let's come up with another plan.
Yeah, good answer.
Yeah, I have one final question, but I'm going to hold it till the end. It's about the recording. So.
Oh, okay, perfect. So Chad, if I see that we're coming to the end, if you'll flip to the last page, perfect. So I know for all of our listeners and our watchers now and on replay, it's really difficult. Oliver kind of alluded to it at the top of the webinar. I've mentioned it a couple of times. It's really difficult to find where all of these clinical trials are housed. And if you're a patient or you know of someone, a loved one and is a patient or impacted by cancer, you're like, where do I go to educate myself and even get an idea of all that's out there, or even providers? And so let's talk to these two, because I am particularly in love with theranostictrials.org. I think Dr. Norquist has done a phenomenal job, but can you speak to how patients, providers can educate themselves and where to go?
Yeah, so you've got two fabulous websites up
Yes.
here. On the left-hand side is clinicaltrials.gov, which is like everything.
Yeah.
Now, the good news is you can do some searches by looking at the condition,
Yeah.
look at the intervention, look at the location, you kind of narrow it down. But it's like the whole shebang.
Everything,
It's
yeah.
everything. And it can actually be a little bit confusing at times.
I agree.
And the theranostic trials, and I want to give a shout out to Luke Norquist, who runs X Cancer out of Omaha, Nebraska. He's a theranostic investigator, and what he has done is he decided to aggregate this because there was no aggregation. And he put together this theranostictrials.org, and it is really complete. And
Oh,
so it is searchable as
Wonderful.
well.
It is.
So take a look at that website. I'm not trying to endorse any particular individual. I'm just saying I like that website, and you will too if you're interested in theranostic
Yeah,
trials.
I agree. I think the website, it's not only where, I think clinicaltrials.gov doesn't offer that educational piece as much. Luke's site offers that, whether you're a provider, a patient, or industry, even manufacturer, it offers that. And again, I'm not trying to endorse any particular, I just really want to make sure that people can arm themselves with resources to educate themselves, and that's it.
Information is power.
It is.
And for the patient to be able to understand, or for the investigators to be able to understand, or anybody else who's interested to be able to understand, information is our currency for understanding. And first of all, you have to get the information. If you're looking across the entire world, then you need some organizational structure in order to help you kind of be able to find what you're looking for. I mean, you could Google it, but then the first thing you get is all
That's
the
right.
sponsors, and the
Yeah.
second thing you get is just a bunch of hodgepodge. It's better to have it organized. These are two good sites, and Theranostic Trials is the one that really is focused on theranostics.
And if there are patients or folks online who are maybe more in that regional community setting, I would recommend you tell me, you react to my recommendation, that if you're on the site and you find some clinical trials that you think, wow, I think myself or someone I know could be eligible, take screenshots and take it to your doctor.
Absolutely.
And say, hey, do you know of this trial? What do you think of this trial?
Yeah, very reasonable. Now, the only thing that's a caveat there, it's really hard for a lot of the community doctors to keep up.
Yeah.
And they may not know about the latest PSMA
Yeah.
lutetium trial. And so, at times you may have to call the trial site and spend a little extra time in educating. But that's time well spent.
Sure.
If you have a need, you want to be able to get to the right spot. I will say in general, that if you get to a major center where you have specialists in your particular cancer, those specialists are going to understand sort of the menu of available trials and
Okay.
what's potentially suitable for you, as well as the standard of care options that may not be fully discussed otherwise. So,
Okay.
finding somebody who knows how to treat the cancer that you have, that's really important.
Yeah, really, really important. Fantastic. All right, Chad, I think we have 1 minute left.
Yes,
Did you
so
answer the question on the last webinar, WebEx?
I stopped sharing, so the last question was about the recording of the webinar. Once the editing is done on the video, it will be hosted to Nucleus's LinkedIn website. Also, when you registered, you'll get a follow-up email thanking you for your attendance and registration for the webinar. You can email me; it will be attributed to me. So if you don't see it on LinkedIn, feel free, shoot me a direct email, and I'll get you a copy. Thank
you, Chad. So we have come to the end of our webinar. Oliver, I want to thank you for your time, for just the vast insights and experience that you have. I want to thank our listeners and our watchers. I really hope that this was valuable and helpful to you, a good use of your time. Feedback is always, always welcomed. In fact, please let us know, good, bad, and ugly, what you liked, any feedback, and if anything you didn't like, that is all about me, not Oliver. So feedback is always welcome. And Oliver, last word to you.
Thank you, Caspi. Thank you, Chad. Thanks for having me.
Excellent. Have a great rest of the day and week, everyone. And until next time.